Namaste students, खुश रहो बैठिए. I JVN Dr. Y. Chandrakala, Assistant Professor, Jyoti Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the fluid mosaic model of a plasma membrane. This model was proposed by Singer and Nicholas in 1972. The lipids are thought to be arranged primarily in a bilayer in which the proteins are embedded to varying degree. The he classified the membrane protein as a peripheral or an integral. The protein varies in size and dissolves to vary the degree in the lipid matrix and are able to diffuse laterally in the plane of membrane. And the entire structure is hence dynamic. In this model, the lipid molecules may exhibit intramolecular movement or may rotate about their axis. So there may be displaced flip-flop movement, including the transfer from one side of the bilateral to the other side. So here in this diagram you can see the plasma membrane based on the fluid mosaic model. Uh, these are the boundary lipids present in the boundary side with intrinsic proteins and the hydrophobic heads. Okay, these are the hydrophobic heads, and here this is the bilayer present with the hydrophobic tails. Okay, and these are the extrinsic proteins present here, and these are the internal intrinsic proteins. Okay, these are the intrinsic protein, and here it presents the extrinsic protein, and these are the hydrophobic heads, and in between this is a bilayer lipids. Okay, this is the plasma membrane based upon the fluid mosaic model. Okay, so here the lipid glycoproteins and many of the intrinsic proteins of the membrane are amphithetic molecules. That means having a liquid crystalline combination in which the polar group are directed towards the water phase and the non-polar are situated inside the bilayer. Okay, so the lipid bilayer forms the structural matrix, so which serve as the permeability barrier of the membrane. Okay, so in this membrane with high lipid content or a bilayer lipid extensive or interrupted only by occasional by a protein molecules. Whereas in the membrane of high protein content, the extent of the lipid bilayer is reduced. Thus, the fluid mosaic model may be described as the chemical composition of a molecules organized and ultrastructure of a plasma membrane. This arrangement allows the various enzymes and antigenic glycoproteins so have their active sites or exposed to the outer surface of the membrane. Here the fluidity of the lipid depends on the degree of saturation of the hydrocarbons or the ambient temperature. Thank you. This session is powered by 2.0 Jyoti Vidyapeeth Women's University, Jaipur. If you have any queries, we'll discuss in the problem solving session.